Hi everybody, welcome to video number 2 on our reproduction unit. This is going to be a video that's going to contrast mitosis with the cellular process of meiosis. Meiosis is a form of sexual reproduction, and its primary purpose is going to be to produce gametes. Gametes are sex cells. They are cells that are specialized to be able to combine with each other to produce a new organism. In this case, these are exemplified by sperm and eggs, or if you're talking about in plants, it can be pollen and ovum. Fertilization is that time period or that moment when the sperm and egg fuse to produce what we refer to as a zygote. A zygote is a fertilized egg that has genetic information from both parents. The process of producing gametes is known as gametogenesis. The term is actually a combination of two: gamete, which is a sex cell, and genesis, which means beginning. The process where gametes are produced occurs in specialized tissues called gonads. The gonads are typically the testes and ovaries in humans, but in plants, this is going to be found in the flower. Hermaphrodites are a unique group of organisms that can be plant or animal varieties. It's not restricted to just certain types of organisms. Lots of different species have hermaphroditic organisms, but they contain both male and female gonads. So, for instance, in our image here, we have some worms. Worms are actually hermaphrodites. They produce male gametes and female gametes. So these two worms are going through a process of sexual reproduction where there's an exchange of gametes. The male gamete or sex cell tends to be motile, which means it can move. So the male gamete is going to be passed from one worm to the next. And the other worm is also going to be passing its male gamete over to the other worm. So they're both going to be fertilizing their eggs in this mating situation. So the question then becomes, what is this process of making sperm and eggs? And we call it meiosis. Some people say meiosis. Either way, it doesn't really matter how you say it. It's more important that you understand the process. So, whoops, sorry about that. Meiosis is a process to make sperm or eggs, and it's often referred to as reduction division. We say reduction division because it's a type of cell division that reduces the chromosome number by half. So what really helps if you're trying to compare the chromosome number and how it's reduced by half, the best way to do that is by comparing the mitosis process that we learned about with the meiosis process that we're talking about. The best way to, so let's take a second. I'm gonna have you draw with me a little bit. There is a little bit of space in the notes, and I'd like you to draw a couple circles. I expect your circles will be as perfect as mine. And then let's label what this is. So we're gonna be drawing mitosis, and the cells in mitosis are 2N, which means they have two full sets of genetic material. You get a set from mom and a set from dad. Well, in meiosis, the same thing is true. All right, you still start with a cell that has a set of genetic material from mom and a set of genetic material from dad. The best way to see how it's actually different then is to actually draw in here some chromosomes. And we're going to use metaphase. And I'm going to put 2 different colors in here just so you can see. If we were to say, all right, there are 3 chromosomes from mom and 3 chromosomes from dad, well, here's how they line up differently. In mitosis, where's my draw? In mitosis, they line up along the metaphase plate. Wow, that was pretty bad. All right, let's try that again. They line up along the metaphase plate, and when they separate, half the chromosomes, half of a chromosome goes that way, half goes the other. The chromatids separate and move towards the poles. All right. In meiosis, however, they look like this. Instead of lining up single file, they line up with their matching pair from mom and dad. And it's not hard to make an association between the mom chromosomes and the dad chromosomes matching up and getting right next to each other, because this is sexual reproduction. So now, because they line up like this, the first division is going to actually cause these chromosomes to not be pulled apart like they were in the original, in mitosis. So if I put the finished product here, all right, let's go like this. All right, and let's put a little arrow in here because we get, oops, not that kind of arrow. How about this kind of arrow? We get 2 cells, and those cells are going to be identical. We're also going to get 2 cells in meiosis, though, also. So we're gonna do this. We're going to show our 2 cells. Now remember, you should be drawing this because it is going to be helpful for you to visualize. All right. Now, in this particular case, you'd get but in addition to those 3, you'd also get 3 that were red. So the nucleus that's formed during mitosis is going to have a set of genetic material from mom and a set of genetic material from dad. You're going to have an identical set of genetic material from mom and a set of genetic material from dad in the other cell. These cells are identical and they have 2 full sets of genetic material. Okay, so let's look at what the outcome is here in this case. When we put this in, we actually end up with, I'm sure that as you're viewing this, you're seeing, oh, wait a minute, that's definitely not right, because that's a full chromosome and its copy, they haven't been separated. So the cell has cyclins that we learned about that also tell it, hey, wait a minute, we're supposed to have separated these. So it's going to go through a second cell division. So in doing that, each cell is going to split a second time because it needs to have just one chromosome. You can't have two copies connected together. So I'm going to run out of space here. So I'm going to make them a little bit smaller. All right. And that second division is going to give you what we were originally talking about. And then over this division on this side, you're going to have and. Well, what have you done to the amount of genetic material in each nucleus? Each nucleus, instead of having 2N, these are going to be N. It's just one set. It's reduction division. You've reduced the amount of genetic material through the process of cell division, and it's all because your cells don't want a full chromosome. They want to have one copy. All right, so that's the signal that tells it needs to divide one more time. And it's the lining up during metaphase that sets that up. All right, so let's look at what happens on the next page. This is going to outline this process again. All right, so all the way through the process of meiosis, it's 2 cell divisions now, just like we had in the picture on the previous page. Prophase 1 is going to show those chromosomes matching up, the mom copy and the dad copy that are going to get together. These are called homologous chromosomes. There's a mom and a dad version. All right, the blue and the green, they're going to match up together. And during this, there's a very unique process that occurs to increase the amount of variety in the offspring. This is called crossing over. Chromosomes can exchange and swap segments that are right near each other because the 2 chromosomes have the same type of information. So there might be a gene for eye color on here, all right, and it's going to swap the eye color gene between mom and dad, giving the potential offspring different combinations of genes. That's what crossing over does. It shuffles the genetic deck, all right, giving opportunities for new, unique combinations of genetics that wouldn't normally happen through mitosis. Metaphase, they line up single file across the metaphase plate. And remember, I taught everybody that it always separates perpendicular to the metaphase plate. All right, there are the centrioles. All right, and there's going to be a perpendicular separation from those centrioles. And because they paired up, the whole chromosomes are going to move towards the poles. And that's anaphase 1. All of the phases, or all the phases in meiosis 1 are going to be labeled with a 1 because it's part of the first division. So the tetrads are the 4 copies that are lined up together. That brings me to that separation, which is called disjunction. There's a disorder that can occur called non-disjunction, and that can lead to different genetic disorders like Down syndrome, when chromosomes don't separate properly and you end up with extra copies. All right, so non-disjunction is when chromosomes don't separate. And that can lead to Down syndrome. All right, there are lots of genetic conditions that result from non-disjunction, but Down syndrome is just one that you're probably more familiar with. Telophase is the first division. We discussed that earlier. The cytoplasm divides, you get 2 cells, but because they have full chromosomes, they've already got an original and a copy with it. It needs to separate again. That means there's going to be a second division. That brings us to meiosis 2. Meiosis 2, the chromosomes don't duplicate. Notice there's no interphase. All right. And one of the things that happens during interphase is the S part of interphase is when all the DNA is duplicated. There's no S phase, no duplication. It's going to go right into the division again. That's going to bring us to prophase 2. All right, chromosomes are present. They're going to move towards the middle. That's metaphase 2. And you'll notice that in metaphase 2, now it looks like regular mitosis. They're going to line up single file again. All right, so you've got a single file line, and they're going to separate towards the poles again. The centromeres divide and split. The chromosome halves, the daughter chromatids. That's remember that. That's all right, these are daughter chromatids. Are going to move towards the poles. Now, once they separate, they're not tetrads, all right, and they're individual chromosomes that are moving towards the opposite poles, and they're going to continue to move until they reach the poles. It goes through a second cell division, so there's going to be another cell division for each one of these cells, leading to 4 cells. But you'll notice that we're not calling them diploid or 2N. These are now going to be referred to as haploid or monoploid. They mean the same thing. Haploid means half the normal number. It's supposed to have 2 full sets, but now it has 1. So 2N has been reduced to N. That's half the normal number, or mono meaning 1. These are 1N cells, and these are going to become sperm, or they can become eggs, depending on the gender of the organism. Right? Those are haploid cells, and when we get to a comparison of male versus female, we have 2 processes that occur. In males, when meiosis happens, it's called spermatogenesis. That's the genesis or beginning of sperm. And this happens in the seminiferous tubules of the testes. The testes are actually just tubes with somatic cells in them. And those cells are called spermatogonium. These are the starting cells. They have 2N, but we talked about how all that genetic material gets replicated. It goes through meiosis 1, and then it divides a second time. That's meiosis 2, and you end up with a spermatid. All right, or that's immature. The mature version is a 2N sperm, and you get 4 of them. Each one of them is unique because of the crossing over experience. Now, that's different than in females, however. Females go through meiosis and produce 4 cells, but though not all 4 cells are going to become eggs. So there's a couple different differences between males and females. You definitely want to know the difference between spermatogenesis and oogenesis or oogenesis, depending on who you talk to. But it's easy to remember O and ovaries. all right, they produce eggs. This also is going to have two divisions. So you're going to have meiosis 1. The first division is going to lead to the cells having those full chromosomes. They have to separate. All right. That's going to give you the individual halves that you're looking for. All right. And instead of having 4 N cells after second division, you're going to have 4 N cells, but only 1 mature ovum or egg. The other 3 are going to become polar bodies. These are going to be relevant and important in hormone production to help the female's body produce estrogen and progesterone to manage pregnancy. All right, so that brings me to fertilization. And the fertilization event, if I clear my drawings here, the fertilization event is the fusion of an ovum or egg and sperm. N plus N gives you 2N. So the final product after fertilization brings you back to a 2N somatic cell that can divide by mitosis. From now on, for the rest of its life, it's going to do regular mitosis until it starts to make its own gametes. So those cells are going to divide, and those will divide until the body is produced. And that means the remainder of the cell division, once fertilization occurs, is done by mitosis. That mitosis, remember, is used to grow, develop, and repair tissues. All right, so it's important to remember, I can't really hide that anywhere. There's a difference between mitosis and meiosis and when it's used. So mitosis is used to grow your body. Meiosis or meiosis is only used to make the gametes. That's the only time it's used. Now, moving onward, actually, that fertilization can occur in different places. In humans, fertilization occurs internally. That means, and actually it's typically in the fallopian tubes or the oviducts. But some organisms do external fertilization. That means they have to lay eggs outside of their body. And many cases, in order to survive, these animals or organisms have to produce high numbers of eggs for survival's sake. Examples of organisms that lay eggs, they have to be fertilized by the males outside of the body, include aquatic vertebrates like fish and frogs. They lay clutches of eggs in ponds and waterways, and males will actually come up to them and release their gametes in the vicinity of the eggs and just hope for the best. External fertilization is not really efficient. Internal fertilization is done with organisms that have limited access to water. And so, as you probably realize, sperm need to be able to swim. And so if you have limited access to water, you need to have a moist reproductive tract in order for those sperm to be able to move. So terrestrial vertebrates like humans, or all mammals for that matter, aquatic or terrestrial, all of those do internal fertilization. But there are other organisms that do internal fertilization that are not highly developed. Even some of the insects do internal fertilization. The only other thing that's really relevant to talk about is where the organisms offspring develops. So then you get a comparison of internal development, like mammals. The female grows the baby inside of their body. So that would be an example of internal fertilization and internal development. But there are organisms that do internal fertilization and partial external development. So laying eggs like a bird. Birds do internal fertilization, but they lay an egg and the offspring develop outside of the body. So internal fertilization, external development. And if you have external fertilization, you're going to do external development also, because most of the time there's so many eggs that have been produced. They're all on their own. They're just hoping for the best. All right, if you have any questions about meiosis, feel free to contact us, and I hope you have a wonderful vacation.